Howdy folks, and welcome to this microbiology video where we're going to talk about antibiotic sensitivity with different sets of antibiotics. Now first, to do that, we're going to need to create a LON. Now this is similar to but different from our serial dilution spread plate that we've done previously. The key difference is that we're not going to dilute out our culture that we've made. The other thing for this activity is that you're going to be working in conjunction with your whole bench, all right? So you and your lab partner are going to be working with one culture of E. coli, for instance, and your other lab bench pair is going to be working with Staphylococcus aureus, all right? But for the most part, you're doing the same activity with the same antibiotics. We're just assessing how these different bacteria respond to the different antibiotics. And you're going to be working together as one whole bench to achieve that goal. Now for that, to start, we want to make sure our plate is properly labeled, okay? So first, let's get it labeled. Initials, date, our media type is Mueller-Hinton. It's a media type that we like to use for antibiotic sensitivity tests. For this video, we're working with E. coli, so EC, and then we're also growing this at 37 degrees, and then your lab ID, whatever that may be. All right? And then along with getting this label on there, you're going to divide your plate into quadrants. The reason for that is you are going to be using all 4 antibiotics on the same plate. And because of that, we're going to want to get them labeled accordingly. So we're working with 4 different antibiotics. We're working with chloramphenicol, so you can just label that C, because that's the letter you see on the antibiotic disc. Working with gentamicin. Gentamicin is abbreviated as GEN. We're working with tetracycline, which is abbreviated TE, and we're working with the tried and true familiar penicillin. All right? So label them accordingly. Keep the labels small because you want to limit how much the marker is going to get in the way of your assessment next week when you come back to assess your results. All right? So now we've got our plate properly labeled. We're going to flip it over and get ready for the LON. Just like with the spread plate, we're going to sterilize our spreader in the same way. Dip it in alcohol, and flame it. Now, keep in mind, for safety reasons, you want to keep this near the flame, away from the beaker of alcohol, not waving it around, dropping flaming alcohol all over the place. And you want to wait and make sure this completely cools. A few of you ended up killing your bacteria because you didn't wait long enough and you didn't have anything to work with your spread plates. So make sure that you wait long enough. While we're waiting, we're going to be very careful and not contaminate our spreader, but we're going to go ahead and open up our sterile transfer pipette. Get the transfer pipette out, and then we're going to get our tube, flick it a little bit to get the culture around, open it up, wave the tube opening over the flame, collect a little bit of culture, wave it over flame, cap, and now you're done with the broth tube. You're going to add just 2 drops to the middle. Anything that you don't use, we will simply get rid of in our biohazard waste. Be careful moving your arm over, watch for that flame. Pay attention. At this point, hopefully this is cool, but if you're unsure, it's always better to wait longer. Again, before you touch your bacteria, it's always best to touch the edge of the agar first, and then bring that in to your bacteria before you start to spread it around. Now, the critical thing about this test where we are assessing antibiotic sensitivity is we absolutely need as much bacteria covering this plate as possible. We want a thick, full lawn of bacteria. So you want to make sure that you get this spread around as evenly and as thoroughly as possible. Okay? Recover and let that bacterial broth that we've added to it soak in. Give it a minute. We're then going to sterilize this, dip it in alcohol, inflame it. We are now done with the spreader at this point. Set it aside. Now, while we're waiting for that to soak in for a little bit, you see here these little tubes. These tubes each contain little discs of antibiotics. This is chloramphenicol. We have penicillin here, tetracycline, and gentamicin. I've already listed those off as we were labeling our plate. Each of these contain a disc. These little discs are soaked in antibiotic. That antibiotic disc is what we're going to be placing onto this plate, and over time, over the weekend, as your culture is growing, you're going to have that antibiotic slowly diffusing out away from that disc. And depending on how close bacteria are to that disc will determine how concentrated that antibiotic is. The higher the concentration, the more effective that antibiotic should be. So based on where the growth of the bacteria occurs in proximity to the disc will tell us how effective that particular antibiotic is against that particular species of bacteria. Okay? Now, you are not going to be given these tubes of antibiotics. The reason for that is they're kind of difficult to get the discs in and out of effectively. So what we're going to do is we're going to provide you a small petri dish with just enough discs in them for you and your lab bench. You should see 2 in them for your whole lab bench because both sides, one's working with Staphylococcus aureus, one side's working with E. coli. Both bacterial species we're going to be testing with the same antibiotics. So there should be 2 discs of each type in this small dish at your bench. Before we move these onto our petri plate, we want to sterilize our forceps. So just like with the spreader plate, we're going to dip it in alcohol and flame. The reason for this is we want to make sure that anytime we are contacting our antibiotic, we are not already adding culture there that may not be what we're wanting to assess. Okay? So, let these cool. You don't want to burn your antibiotic disc or the bacteria that you might potentially come in contact with when putting the disc on the culture. So give it a little bit of time. And then at this point, you are also going to want to make sure that you're working close within the flame zone of inhibition, because we're going to have to take off this lid and leave it off for a good little bit. Okay? So, forceps should be cool. We're going to come in, and I'm going to start off with gentamicin, because that's right here. Just grab it on the sides like this. Open the plate. Pay attention to the label. Find your GEN, and you're going to drop it right in the middle of that quadrant you made. Now, the thing is this disc can fall off, especially if we are growing it as we should and turn the plate upside down in the incubator. So to make sure it doesn't fall off, we want to make sure we tap that antibiotic disc into the agar so that it's nice and stuck in place. So tap it a few times, but don't push it in. Small taps are all you need. Go ahead and cover this while you sterilize your forceps. Now, if you are using proper sterile technique, you probably don't really need to flame sterilize your forceps every time, because you shouldn't be touching the bacterial culture on the petri dish, because you should only be touching the antibiotic disc when tapping it into place. But just in case you make an error, always flame sterilize in between antibiotic discs, just in case you did touch culture. Okay? So give it a chance to cool off, and then let's go ahead and now get penicillin. It has a P label. Find your P label on the bottom of your plate and drop it in its respective quadrant. Okay, tap it in. Notice that this is not exactly in the middle of this quadrant. Even though I divided this out 4 by 4, my lines are not exactly even, my quadrants aren't even. So I'm adjusting to make sure that my penicillin is not too close to the wall. You want to give them space to spread out, okay? So use your lines as general indicators, but make sure that your 4 discs are relatively evenly distributed across, okay? Flame sterilize. And again, be careful, don't burn yourself or drop flaming alcohol on anyone. Let this cool. Next, we're going to take our chloramphenicol. Has a label of a C. I'm going to just drop it right in the middle of its quadrant. Tap it down. Flame sterilize. Let it cool. And then add your last disc. In this case, tetracycline. Add it to the middle of its quadrant area. Tap it in. And end with some flame sterilization. Okay. Cover your alcohol beaker. Turn off your Bunsen burner, you're done with it at this point. And then you're going to incubate this at 37 degrees over the weekend, and you're going to come back and see a lawn of bacteria with spaces of no growth near that antibiotic disc. To measure that, you're going to get your handy dandy ruler, and you're going to determine the diameter of that zone of inhibition growth. And you're going to record that, okay? And based on how large that zone of inhibition is, tells you how effective that antibiotic is against that particular species of bacteria.